The 3D organoid system is considered more physiologically relevant than 2D assays, and it can provide valuable information about the biology that would otherwise be challenging to acquire.
This is an adaptable system where we can test pharmacological and genetic manipulations with less time and a significantly lower cost than using an in vivo approach.
Matrix gel is very tricky to handle, as it solidifies when it reaches room temperature. It can only be drawn through a pipette when it is liquid and must be maintained on ice.
The integrity of the ring is important to maintain. If the structure is disrupted, that can negatively affect organoid growth, and you can easily lose material when changing media.
Begin this procedure with preparation of mouse basal and luminal prostate epithelial cells as described in the text protocol. Wash the cell pellet in 500 microliters of mouse organoid media. Then resuspend the pellet at a cell density of 1,000 cells per microliter. To prepare master mixes, mix epithelial cells suspended in mouse organoid media with matrix gel to generate a final mixture that contains 25% cells in media and 75% matrix gel. Depending on the downstream application, basal cells are typically plated at a concentration of 100 to 2,000 cells per 80 microliters, whereas luminal cells are plated at a concentration of 2,000 to 10,000 cells per 80 microliters. For each cell mixture, add 80 microliters of the matrix gel per well of a 24-well plate. Pipette a droplet onto the lower half of the wall of the well, while avoiding direct